At Collaborations Pharmaceuticals, we use real drug discovery intelligence alongside artificial intelligence to develop clinical candidates for rare, neglected, and unmet therapeutic needs. Our core expertise extends to high-quality data curation and machine learning model building, which we offer as a service. We can apply our technologies to drug discovery and ADME tox predictions. We can also conduct in vitro testing in our own labs and coordinate in vivo testing through our collaborator network. We have utilized drug repurposing to get us quickly to promising molecules, as we have demonstrated for Ebola, Chagas, and Alzheimer's disease. And we can apply these approaches to virtually any disease. We are unique in that we publish our own results in peer-reviewed journals and share our data models, unlike other artificial intelligence companies. Please get in touch to learn more about how we can help you at collaborationspharma.com.
Hello, I'm Sean Ekins, CEO and founder of Collaborations Pharmaceuticals. Thank you for joining me today to learn about what's on the horizon, our pipeline update. Before I get into the presentation, I'd just like to mention a few things for those joining live today. I have a few public service announcements. If you would like to ask questions at any time, please click on one of the icons shown here on the slide. You may also connect to us through the attendee chat functions on the left-hand side of your screen. And if you'd like to keep it private, just put that in the message as well. And so I'll be happy to answer questions at the end. If you want to put your questions in there as you're going through, that's fine. But I will be answering questions finally at the end. For those of you that are watching this on demand, we're going to be providing information in the screen as well for those listening live. But if you need more information afterwards, please get in touch and we'll provide that to you. So I'm going to give you just a brief overview to orientate you as to what I'm going to present in the next 25-30 minutes today. I will provide an introduction on the company, basically who we are and what we do. And then I'll go through some of our pipeline assets, and I'll go into a very high level. And finally, I'll just have a summary at the end and answer questions. If you'd like to learn more about some of the assets that we have, please get in touch with us, and I'll have that information at the end as well. So just to begin with, who we are at Collaborations Pharmaceuticals. I founded the company in 2015. We currently have 9 employees that are shown here on the screen. And we're located in Raleigh, North Carolina. We have 3 laboratories. One of these is our BSL-2 lab, where we do in vitro experiments and cell culture. And we're situated in the NC State incubator here on the campus. To date, I funded the company primarily through grant funding from the NIH and DoD, as well as supporting the company with fee-for-service collaborations. I believe that we're a robust company and we have a solid molecule and services pipeline. And I think that this will make us a stable partner. So I'm excited to share with you some of the things that we've been doing on our molecule pipeline today. But first, just to give you a little insight as to what we do at the company, we use and leverage my 25 years plus of drug discovery experience alongside software that we've developed, which is in the area of artificial intelligence. And we use these combined to help develop and identify molecules. And we focus primarily on rare, neglected, as well as unmet therapeutic needs. So we are essentially a drug discovery company, but we do have the capability to perform contract services for you if there's an area of particular interest. And our focus is primarily on early drug discovery. So we have a lot of expertise in hit to lead and candidate optimization. And we have particular expertise in toxicology and ADME modeling. And our focus is really on curation of data, building the models through to performing the experiments. We generally have to tackle some quite challenging problems for companies that we work with. So if there are particular projects that you would like to outsource, please get in touch. We also have a very wide collaborator network as well that helps us in our drug discovery, and obviously we can tap into this as well as needed. So just a little overview of our company pipeline. As you can see, we have quite a large pipeline of molecules that are in different stages, mostly in preclinical. We have a number that have progressed quite far along, and I'm going to try to give you some highlights of these today. As you can see, we have a very heavy emphasis on infectious diseases as well as rare diseases, and that's really our sweet spot. Many of these projects have been funded through NIH or DoD funding, and we've applied machine learning to probably 3/4 of these projects, either for identification of the molecules themselves or for lead optimization. And we have IP in many of these areas through patents or orphan drug designations from the FDA, as you can see on this slide. Our goal really is to de-risk these projects and then find partners to take them into the clinic. And as you can see, we have a number of projects that have reached that stage, and I'm going to describe most of these today and give you a little bit more insight into where we are. So without further ado, I'd like to kick off with some of the diseases of interest to us. And one of these is Ebola. And this is a disease we've been working on for over 6 years, the company. This is obviously very well known because it gets in the news every few years when there's a particular outbreak. What is quite important is the high fatality rate of this virus. And obviously, this is generally focused to countries in Africa, although occasionally there are individuals outside of Africa that are identified with it. And some of the symptoms associated with it seem quite mild initially, such as fever or muscle pain, but this quickly escalates to vomiting, diarrhea, organ failure, and then death in many of those that are infected. And I previously got involved in working on Ebola in the last really major outbreak from December 2013 to January 2016. This outbreak killed over 11,000 individuals and infected many, many more, on the order of over 20,000 individuals, I believe. And since then, there have been a number of other outbreaks that are slightly smaller, 2018 to 2020. There was over 2,000 people died again. And then most recently, over the last year, there's been an outbreak in the Congo. Over 50 people died. And then most recently in 2021, 10 deaths. What's quite interesting about this is that over the years, there's been the development of both a vaccine and a monoclonal antibody, which are now approved, FDA approved, and are available. But there is still a need for a small molecule treatment for this virus. And some of the challenges that are inherent with this particular virus is that we're still learning about infection. And most recently, some of the outbreaks may be due to latency of the virus in previously infected individuals. And this may also be sexually transmitted. And so this may be something that's quite challenging. So there's definitely a need for small molecules to treat this that can be readily applied, and that they need to be shelf stable and cost effective. So those are some of the areas that a small molecule would be able to address. And as I mentioned, our initial foray into working on Ebola came about through machine learning. And back in 2014, I obtained some high throughput screening data of molecules screened against Ebola virus, used these to build various machine learning models, and then performed a virtual screen. And these were used to identify 3 molecules, tellurone, quinacrine, and pyrinaridine. And all of these were initially tested in vitro against the virus in HeLa cells and found to be active in the nanomolar area. And subsequently, we've been able to obtain funding and work with collaborators at SRI to perform animal studies. And some of these animal studies were performed at UTMB, and collaborators there were able to demonstrate that we could show virtually 100% survival in most of these cases, and in the case of quinacrine, 70% survival in the mouse-adapted model of Ebola infection. And we've published all of this work over the last couple of years. And so this was quite promising for us to demonstrate that we could go from a machine learning model through to in vitro, all the way through to in vivo efficacy of molecules. Another point to mention is that all of these were pre-existing drugs approved somewhere in the world. And so this is a really nice example of drug repurposing. Over the last year alone, we've added considerably to our knowledge of the mechanism for these molecules. We've shown that all three molecules are active against three different Ebola strains with activities, IC50s around 1 micromolar, and also are active against two Marburg virus strains with similar levels of activity. We've been able to quite extensively characterize their in vitro mechanism, and we believe this is due to the lysosomotropic nature of the molecules. And in particular, pyranardine is shown to be a 560 nanomolar lysosomal inhibitor. We've also demonstrated that these molecules can inhibit the pseudovirus. For example, pyranardine inhibits the pseudovirus for Ebola glycoprotein with an IC50 comparable to the IC50s obtained in wild-type virus. And we've also demonstrated that these molecules can bind to the glycoprotein. So, for example, I'm showing here the Ebola glycoprotein binding is about with a KD of about 7 micromolar. And then most recently, we've also demonstrated that pyranardine, when given orally to guinea pigs, can also increase the survival. And so we demonstrated 40% efficacy in this lethal model. And what's challenging about the guinea pig model is to date, there have been no small molecules that have been able to demonstrate survival. And so we were able to demonstrate a small but statistically significant survival in this model, which is quite an achievement. Currently, we have pyranardine and quinacrine being tested in the Marburg mouse model at UTMB. So we're awaiting the data. Hopefully that will demonstrate some efficacy against this virus in vivo as well. We believe that there is a clear pathway to the clinic, especially for pyranardine, because we've got so much data that we've accumulated, but also because the effective dose that we're seeing in the mouse can be scaled to a dose that has been demonstrated in humans previously in clinical studies. So this, we believe, is quite viable as an approach. I think the next stages here are obviously to test in non-human primates and to try to see if we could use the FDA animal rule. To date, we have considerable intellectual property on this particular area. We have 3 orphan drug designations for these molecules for Ebola, and we've also filed a patent for use against Marburg and other viruses. Obviously, this ability to have a number of antivirals in our pocket put us in a pretty good position with the COVID outbreak over the last year. And so we were excited to test these with a number of different collaborators against various cell types that were infected with the SARS-CoV-2. And this work has been recently published this week, actually. And I invite you to take a look at this publication online, which demonstrates that pyranardine, quinacrine, and tolurone can show comparable IC50s against SARS-CoV-2 when compared to remdesivir, which is in the low hundreds of nanomolar. So this is quite exciting. We demonstrated this activity in a very relevant cell line, the A549 plus ACE2. And most recently, we've also shown with collaborators that these compounds are active against the mouse-adapted SARS-CoV-2 as well, and again, on a par with remdesivir. So we believe the next step is to really test these molecules in the mouse model of infection, and these may be viable molecules for taking into human clinical trials if they demonstrate efficacy. So we've essentially taken these molecules from Ebola to Marburg and now to COVID-19. I'd just like to move on to a different disease now. This is a disease that's spread by a parasite that's called Chagas disease, and it affects anywhere from 7 to 8 million people worldwide. And it's spread by a triatome bug that carries the parasite Trypanosoma cruzi, which essentially causes the disease. And initially, this seems quite a mild disease where the individual is infected, there'll be swelling, and it will result in a mild fever. But over really a series of years, in the order of decades, it will result in congestive heart failure, which obviously is fatal ultimately. The disease itself is curable if the individuals are treated soon after infection. And in the last couple of years, there have actually been 2 drugs that have been FDA approved. However, due to the toxicity, they're only approved for children from the age of 2 to 18. So there is a dire need for drugs to treat the chronic disease in adults that are identified with Chagas disease. And at about the same time that we identified pyrinaridine as a molecule with activity against Ebola, we also identified it as an antiparasitic against the Trypanosoma cruzi, or the T. cruzi. And we showed this in vitro and also in an infection model in the mouse. And this work was published in 2015. And since then, we've been progressing this into the chronic mouse model and doing additional experiments. And so we've recently finished this, and we're undergoing the data analysis for the individual single treatment of pyrinaridine, as well as a combination of pyrinaridine and another drug. And to date, we've been able to obtain 2 FDA orphan drug designations for pyrinaridine in this particular disease. We believe that we're also well set up for the next stage, which would be a clinical trial in Brazil. And we have a number of collaborations in place there. So this will obviously hinge on the successful data if we obtain that from the chronic mouse study. But again, this is a late stage molecule pretty much ready to go into a clinical trial. I'm just going to shift tack slightly into a bacteria. We've been working for over a decade on tuberculosis. And this is an area where we have quite a number of publications. It's obviously a key disease globally. It results in close to 2 million fatalities every single year. And obviously, tens of millions of cases are documented every year as well. What is interesting here is that for many decades, there have been highly efficacious treatments for TB. However, they suffer from really low efficacy. There's also a long treatment duration on the order of 6 months for drug sensitive and 18 months for drug resistant TB, if not longer. So that there are a relatively limited pipeline of drugs going into clinical trials. But most recently, a new class of molecules has been identified that goes after a new target. And these molecules are shown here. These are members of the benzothiazanone class of drugs. They're both currently in phase 2 clinical trials in Europe. One of these molecules is called BTZ-043, and the other has a name called mecozinone. Both of them go after a target called DPRE1, which is an essential target involved in the maintenance of the cell membrane. And we've recently identified a new molecule that is a benzothiazanone. However, unlike the molecules in clinical trials, which have very, very poor solubility, our analog has a high solubility, and it also maintains the in vitro activity, so in the order of low nanograms per mil activity. And we've also demonstrated that it is still on target with DPRE1. We've undertaken some preliminary in vivo efficacy studies showing that this particular molecule that we've called 119-26289 demonstrates a 3 log CFU drop in infection in lungs of mice. And we're currently having a larger study ongoing to test the in vivo efficacy. And then the next stages will be to do pre-IND type work and to move this into a clinical trial. We believe that this is an improvement over the prior molecules, and having the increased solubility will make this more useful when it comes to making formulations that are acceptable. Another area that we've got considerable expertise in is HIV. Again, this is a disease that still kills close to 2 million individuals globally. It obviously affects a very large number of patients in the US as well. On the order of 40,000 people are still diagnosed every year with HIV. It is generally well controlled with combination treatments that are available. However, what is seen in the clinic is that a number of patients have HIV-associated neurological disorders after being on drugs for many years, if not decades. And so we've been thinking about how we can try to address this by going after drugs that get into the brain but are not neurotoxic. And one particular well-validated target is the non-nucleoside reverse transcriptase inhibitors. And to date, we've made over 250 molecules of an original chemical series. And we've identified a number of molecules that are now down into the sub-nanomolar region. And we've tested molecules against wild type and mutant HIV. And one molecule is shown on this particular page, for which we have low nanomolar binding, and we have really good ADME properties and PK properties shown on the right-hand side. We've taken this molecule into mice studies to look at PK, maximum tolerated dose, and demonstrated that the drug actually does cross the blood-brain barrier. And this is a good representative of the chemical series. And currently, we're undertaking a number of studies. We've just finished performing in vitro studies on neuronal cells, which I'll show you in a moment, at UNC. And we have crystallography studies ongoing. The next steps for us will be to take this model into an infection model, and our goal is to patent and publish this work later this year. And so just to give you an idea of how a number of these molecules perform, both in vitro, with activity against the whole cell virus, and then also against primary mouse neurons. I'm showing you some of this data here. As you can see, we have a number of molecules that are sub-nanomolar, that demonstrate relatively low, if any, toxicity against these cell types. And a couple of examples are shown on the right-hand side. You can see that the well-known NNRTI efavirenz is neurotoxic, as it's well recognized. And our compounds certainly don't show anywhere near that level of toxicity. And you can see that also on the table on the left-hand side for a couple of our compounds. For example, 120-26124, we have sub-nanomolar HIV activity, and we have a very clear separation, no neurotoxicity. A more sensitive marker for neurotoxicity is intracellular calcium accumulation. And you can see here that there's a spike in calcium accumulation for efavirenz, and our molecules are essentially around the baseline. So this bodes quite well, I think, for demonstrating our molecules may be very useful in this particular aspect of the disease. I'd like to shift direction slightly and discuss a couple of rare diseases now. And one of these that we've focused on for a number of years is Batten disease, or CLN1. And this is an important neurological disease that ultimately is fatal to the children that have it. There are a number of symptoms in which the disease is readily recognized. The children have vision issues, and they have mobility issues initially, and then the disease rapidly progresses, and ultimately it leads to their death. And currently there are no treatments for this rare disease, and with colleagues in academia, we've been involved in pushing forwards an enzyme replacement therapy using the human recombinant protein. And although there have been some earlier in vivo studies in mice showing that it can slightly prolong the survival of the animals, we've been interested in looking at a couple of different dose routes. And so we've recently finished with collaborators performing an ICV, or intracerebroventricular dosing, and intrathecal dosing, as well as a combination of these dose routes in mice. And we're currently analyzing the data, and we have a paper in preparation. What I'm showing you on this slide is a couple of images where we demonstrate that the particular ICV route does very well in looking at a couple of markers for storage of the lysosomal enzyme. And you can see here that the animals that have been given this particular protein, they have levels of these markers that are comparable to the wild type that's shown on the left-hand side. We've also demonstrated that we can put about 30% of the wild type level of enzyme into the brain using the ICV route. Generally, anything above 10% is thought to be acceptable. And we've been able to decrease some of the secondary enzyme markers like beta-glucuronidase by dosing the animals through this particular route. We have data that we're currently assessing for rotarod, as well as for the animal's ability to walk. And so we'll be looking at the gait analysis from these studies and seeing if we can show some improvements in these phenotypes as well. In terms of intellectual property, we have the orphan drug designation and also the pediatric rare disease designation already in hand. And I believe we're well set up to file a pre-IND. And we've also identified clinical manufacturing and also sites for toxicity, as well as clinical sites for a small clinical trial. And most recently, we submitted a phase 2 SBIR through NINDS, and we're just awaiting information on that. I believe that we are in a good position to potentially take this protein through clinical trial and obtain a pediatric rare disease voucher in the next 3 to 4 years. And obviously, we would need to do this with close ties to the patient community, which we have. And the patient community is relatively small in the US, in the order of 20 patients. So the clinical trial would likely be very, very small. And this obviously would keep costs down. Another rare disease that we've been working on is called acute flaccid myelitis. This is also ultra rare in that it really affects maybe a couple of hundred children every year in the US. And it is associated with a virus called enterovirus 68. This is thought to be one of the causes of this particular disease, which is very much like polio in that the children have very serious neurological effects. They're basically paralyzed for their life. There is no treatment for this disease, and we're not aware of any other companies currently that are working on this. Previously, it was thought that another enterovirus compound called pleconaril might be of use, but this has not demonstrated efficacy in any models of the disease to date. What we have shown is an analog of this molecule we've identified as 10 times more potent against enterovirus 68. And we have activity around 20 nanomolar in vitro. And we've also been able to design out some of the issues that pleconaril itself has. So we've been able to remove 3A4 induction in hepatocytes. There is no pregnane X receptor induction. We've also demonstrated that this molecule has good mouse maximum tolerated dose data, and we've shown good PK. And most recently, we've completed an in vivo model with a mouse, and we've currently got a second efficacy study underway. We also are collaborating with an academic group to generate the cryo-EM data for these analogs. And we've shown in preliminary studies so far that the virus is pretty potent at blowing the protein up. And we think that this is because it is binding to the capsid and maybe destabilizing the protein of the virus. We've submitted a patent on the molecules that we've developed, and we've published some papers in this particular area of research. What's really interesting about this molecule is it is very potent against enterovirus 68, but it also has good activity against a number of other enteroviruses, such as Coxsackie. It also has activity against polio, which I mentioned, and other enteroviruses, such as enterovirus 71. So there may be broader applications for this molecule. It also, I should add, has activity against a number of rhinoviruses, which is the common cold, which is generally quite common in that it affects millions of people every year. I have a little bit of data to show you from our recent in vivo respiratory model with this molecule. What we have is a little bit of data showing that the levels of virus titer go down at the 30 mg per kg dose. And this is statistically significant, and it's on a par with a positive control called guanidine. And we've also demonstrated a number of cytokines are affected by the drug treatment as well. And one of these is RANTES, which is an inflammatory marker. And you can see that even on day one, the molecule is able to statistically significantly reduce the cytokine level down to the control levels. So it's on a par with the control. So this is very exciting and would represent, obviously, a novel treatment for an ultra-rare disease, which I believe the NIH would be very excited about because they've written a number of papers in this particular area over the last few years. A further rare disease that we've been working on is Pitt-Hopkins syndrome. This is a neurodevelopmental disorder. These children have essentially a developmental delay. They have issues with breathing, epilepsy, as well as GI issues. And probably most notable, they have a lack of speech. It affects over 1,000 children globally. And just a few years ago, it was identified that transcription factor TCF4 results in basically an intrinsic excitability of the prefrontal neurons. And a couple of targets were also identified. One of them was a sodium channel, NaV1.8, and the second target was a potassium channel, Kv7.1. And it was demonstrated in vitro that basically these deficits in excitability could be rescued by using antagonists. And the antagonists that we used were really just model compounds and not really drugs. And what we have gone and shown is what I'm going to highlight in the next couple of slides is that we've been able to repurpose some FDA-approved drugs for this particular disease. And this was performed with collaborators and essentially the results of a high-throughput screen of FDA-approved drugs. And we identified a number of calcium channel antagonists that demonstrated NaV1.8 activity. And we're showing some of these IC50s here. And of particular interest is one of these, nicardipine, which is submicromolar activity. We demonstrated nicardipine has an IC50 of 0.6 micromolar when we use the patch clamping approach. We've also taken this molecule through an in vivo model. And so we've used a well-characterized knockout mouse model for Pitt-Hopkins syndrome. We used a dose of nicardipine of 3 mg per kg once a day for 3 weeks. And we looked at a number of measures of the phenotype, including sociability, nesting, fear conditioning, self-grooming, open field test, as well as test of force. And what was seen in all 6 of these measures was that nicardipine-treated animals essentially returns the knockout mouse to wild type in terms of these phenotypes. So this was very exciting, and I believe this is the first time that anyone has ever demonstrated that a drug can reverse the phenotype in the mouse model. So this is a molecule that is a repurposed drug. We obviously know that this is available and it's used widely orally and IV as a drug to treat high blood pressure and angina. What's interesting about the molecule is that it generally doesn't decrease the blood pressure in normotensive individuals. So this would not be a concern when treating individuals with this drug. And This molecule is also known to cross the blood-brain barrier, and it has been evaluated in clinical trials for various other CNS diseases. And this drug, in our opinion, is really ready for a clinical trial because of what we demonstrated. And as a proof of concept, this is something that the population of individuals with Pitt-Hopkins syndrome are obviously waiting for. Currently, we have the orphan drug designation and the rare pediatric designation for nicardipine for use with Pitt-Hopkins syndrome. And we've published 2 papers on this particular repurposing project. The final example I'd like to share with you today is our work on Alzheimer's disease. Obviously, we could probably have a full presentation on Alzheimer's disease and the many failures that have occurred over decades. This is obviously a massive disease globally, and in the US alone, it affects nearly 6 million people. There is currently no cure, but there are a number of treatments that have some effect on the disease, maybe slowing it slightly. And these include the acetylcholinesterase inhibitors, which is a valid target. These have some issues themselves in that they have some side effects. And this may be due to the potency. And recently, with a repurposing screen, we identified that a molecule that's already approved, which we mentioned previously, Tilarone, this actually is a potent acetylcholinesterase inhibitor with an IC50 of about 64 nanomolar. And it is also selective in that it doesn't inhibit the closely related butyl cholinesterase. And this information is shown on the right-hand side of the slide. We've also performed a safety screen using the 44 important tox targets, and we've shown that only acetylcholinesterase was really the target of this particular molecule. And we've also tested over 485 kinases and demonstrated no inhibition at 1 micromolar. We believe this is interesting and important because it is a less potent inhibitor of acetylcholinesterase than donepezil by at least an order of magnitude. And this, of course, may be important when it comes to the peripheral side effects of the molecule. We filed a provisional patent for this application already. And this molecule is ready for in vivo studies, and we recently submitted a grant to the NIH to try to see if we could do this. Interestingly, the compound is available already over the counter in Russia and the Ukraine, and so there are a couple of decades of experience with this compound and any particular side effects that it may have. We think that this is an exciting new molecule that could be readily applied to Alzheimer's disease as another potential treatment option for patients. And we recently published this work in Chem Research Toxicology. So in summary, I've highlighted some of our pipeline, which is available for partnering. So if you're interested in any of these particular areas or learning more about them in detail, please get in touch. We have 8 current projects that have completed or have in vivo studies ongoing. So we have been able to generate considerable amounts of data. We also have IP, or we have orphan drug designations for many of these projects. As I described, our rare disease projects generally have a rare pediatric disease voucher designation. And this then obviously allows us, upon FDA approval, the potential to obtain a voucher which can be monetized. And some of these have been selling on the order of $100 million per voucher. I believe that I've demonstrated today some of our capabilities. We can also work on drug discovery projects that you may be interested in. So if this is an area of interest, please get in touch. I'd just finally like to acknowledge funding from the NIH and the Department of Defense, and finally, North Carolina Department of Commerce. So if you have any questions, please put them in the chat box, and I'll be happy to answer them. And I can be contacted also at sean@collaborationspharma.com. And finally, the next webinar, which hopefully will be in April, will be on our machine learning software, Assay Central version 2.0. Thank you for your attention, and I'm now going to take a look at your questions. I don't see any questions today. If you'd like to ask me any questions offline, please get in touch. And thank you for your attention.